Okay, so hopefully everybody's recovered from our last exam. I just want to congratulate everybody. Overall, it looks like everybody did quite a bit better on the second exam than the first exam. I don't know if it's because of my level of transparency, because I give you a week to study, or maybe just the content was easier. It was very straightforward, and maybe that's a part of it. So today, we get to start chapter 12, which is nervous tissue. So I don't know if you can tell, but I am very excited. This is very much my level of expertise, so I'm super excited to talk about this. So I apologize if at times I get going a little quick. I will try to keep that under wraps. And I think that this is going to be a lot of fun. The next couple chapters, especially just again, because I think that this stuff is super interesting. And hopefully you find it interesting too. Okay, so in terms of the rundown, hopefully you kind of have this rhythm in your mind. Your study guide questions should be due for chapter 12 by Tuesday and Thursday. Your quiz is due on Friday. Make sure you have done your pro section by midnight Friday. There are 3 parts to the pro section. Some of them talk about graded potentials, some of them talk about action potentials. I think there's also, there's one other section, I think about neurotransmission. And then also make sure you have completed your adaptive practice for chapter 13 due the following Monday. So again, hopefully this is no big surprise to you and that you're used to kind of just checking on your assignments on Wiley Plus and then doing your study guides before you come to class. Okay, so today we've got 8 study guide questions we're going to cover. Things like talking about the structures and functions of the nervous system, talking about different cell types. Ooh, I don't see gray and white matter, but we'll hopefully discuss that as well. And then talk about cellular properties that permit communication among neurons and effectors. Okay, so the central nervous system. Ultimately, the purpose of the central nervous system is to take, all of the environment around us, take it in through the sensory system, and generate, an adaptive type of response. So it helps us also maintain internal homeostasis. So understand helps us to control conditions which limits and that maintain health and homeostasis. So it does help maintain, all of the different mechanisms occurring within the body. Learn about different branches of the nervous system, and identify various types of cells that are found within nervous tissue. So there are a couple different cell types within the nervous cell tissue. They all have kind of a specific job. And then we'll kind of talk about how there's just 2 kind of general overall types, and then there's subtypes within those 2 types. Okay, overview of the nervous system. Okay, again, if you are a very visual person and you find these videos helpful, make sure you are opening up these PowerPoints and then clicking on these links. Hopefully you have Wiley Plus open at the same time and that you're getting sent to this beautiful video image of an introduction to the structure and function of the nervous system. Okay, so our first image here is this kind of nice overall image explaining the different kind of divisions of the nervous system. And so if we're going to divide the nervous system into kind of 2 different parts, you have the CNS, which stands for central nervous system. And then you have what's called the PNS, which stands for peripheral nervous system. So the central nervous system consists of things like your brain and your spinal cord. Those are kind of all just, I guess, I don't want to say centrally located, but that's kind of like the mastermind, pardon the pun, of the system. So these are, again, where a majority of the functions are taking place. Now, with that being said, I kind of explained how important the central nervous system is to interacting with your environment. And so outside of just that brain and that spinal cord, you're also going to have nerve endings that extend out of your spinal column that are going to interact with your environment. So things like the cranial nerves, if you remember when we were talking about some of the musculature of the eye and of the face, many of those cranial nerves are innervating your face and allowing things like, even as I'm talking right now, it's my cranial nerves that's mediating that muscle movement that allows me to speak. Your spinal nerves are going to, again, extend out from your spine, innervate, your muscles of your appendages and allow your appendages to move. Again, all movement is mediated by excitation of your nervous system into those musculature. Another part of the peripheral nervous system is called the enteric nervous system, or your stomach gut area. It's interesting. It's actually kind of independent a little bit from your central nervous system. They often call the gut the little brain. It has its own kind of set of neurotransmitters, and so it's kind of an interesting part of the nervous system that people often forget about. And then here you have sensory receptors in their skin. So again, important is taking in information from the the world around you. And so the sensory receptors on your skin, all different types of thermoreceptors and mechanoreceptors that allow you to feel touch and temperature are all different types of sensory receptors in your skin. So you'll notice that I'm kind of going into probably a little bit more detail here, just for examples and things like that. But for purposes of class, you just need to kind of know the basics of what's being shown on this slide. Central nervous system is your brain and spinal cord, and then your peripheral nervous system are your cranial nerves, your spinal nerves, your enteric system, as well as the sensory receptors on your skin. So again, getting a little overzealous, but I hope that the examples aid and help in your understanding and aren't a hindrance. Okay, so this is another nice way to kind of divide the functions of the central nervous system and the peripheral nervous system. So we just kind of saw kind of the structural division, like where the central nervous system is located and where the peripheral nervous system is located. But what this figure shows you is kind of how the two interact with one another. So if we look at again, part of the function of this nervous system is to interact with the environment. So here you have the sensory division that has all the sensory input going into the central nervous system. So all of your sensory receptors, whether they be your taste or your smell or your sight or your ears or your touch, all of this fall under kind of special senses. Touch would be more your somatic senses. The sensory division is essentially sending all of this input into your central nervous system. So again, everything that we take in from our environment is being sent to that brain, and our brain is essentially processing it and deciding how it's going to kind of respond to that environment. And so we'll here in a few slides, we'll probably talk about things like the afferent and efferent pathway. So all that sensory information that's being sent up into the central nervous system is going afferent, and then all of the output in terms of responses are efferent. And those two pathways tend to be kind of confusing just because they sound so similar. So I'm just going to repeat them multiple times, and hopefully this motion and me saying it multiple times will kind of embed it into your memory. Okay, so all that sensory input goes to your central nervous system. There are specific regions of your brain that process different sensory systems, and eventually kind of comes together into, okay, we have to create some type of action. There has to be some type of response that's put out to our body to respond to this stimuli. And so you have things like the somatic nervous system and the autonomic nervous system. And so the somatic nervous system, when we're talking about that, we're talking about, you know, our responses going out in form of like emotion. In that case, your somatic nervous system would activate things like your skeletal muscles. So again, like right now, as I am speaking, as I'm gesturing, that is my brain sending efferent signals down into my somatic nervous system and telling me to do all of these things and activating my skeletal tissue, allowing me to move. Now, in contrast, the autonomic nervous system is far more kind of like, I think of autonomic and kind of automatic. You know, those words kind of sound very similar because this is the type of the part of the nervous system that we don't really consciously think about that much, right? You've got your sympathetic nervous system, your parasympathetic nervous system, and your enteric nervous system. So if we think about these things, we don't really have control or really don't cognitively process things like our smooth muscle, our cardiac muscle, our glands. Those things just happen kind of automatically. And so those are all part of the autonomic nervous system. Now we'll maybe further divide the sympathetic and parasympathetic nervous system because they are different. But we're going to, I guess, keep them very general here at this point in time. Again, the enteric nervous system, you know, your gut, you don't necessarily have to think about digesting things. It just happens. And so those are very much the automatic part of your nervous system. So again, somatic, things like musculature, automatic, that just happen as your body is kind of carrying out its regular functions. The things like your heart beating, your breathing, you know, just everything that's kind of automatically happening in the background of your body right now as you're sitting there. Okay, so this is kind of just a rehash of what we just talked about. So your sensory nervous system senses change through the sensory receptors. So think about your senses. You have sight. You have sight, smell, taste, hearing, and touch. And there are also nervous system kind of equivalents to that in terms of kind of more intense vernacular, and we'll get to that at some point. Your motor system, again, you're going to respond to that stimuli, so all that sensory information comes in. And then your motor response is that response to that stimuli, as we saw with the somatic nervous system. And then also another function of the nervous system is to integrative. So you're taking in all of this information, right? Like let's say you are sitting in your dorm room right now and you smell someone cooking something down the hallway, and you're reading your textbook and you're watching this lecture and you're seeing that the light is changing outside. All of those things are happening, simultaneously, and your body is constantly taking in all of that information. And so your brain is able to process it all, essentially create the world around you that you're perceiving. And that's kind of an interesting thought, because we aren't necessarily going to get into consciousness, but it is kind of interesting to think about how our sensory receptors play such an important part in creating our consciousness that we perceive. Okay, so organization of the nervous system. Again, if you are somebody who likes videos, this is a great opportunity for you to check that out. Okay, so histology of the nervous system. So we talk about histology, we're essentially talking about the tissues, the study of the tissues of the nervous system. So if you took a brain, you started slicing it up and you actually looked at all of those tissues, what would all those different tissues look like? What are the types of cells that you find? What are the type of structures? So histology is able to tell us a lot about kind of that structure function relationship of the brain. Again, if you are a video person, you can watch this on nerves and nerves and neurons, as well as neuroglia, but we're going to cover that material here in a second. Okay, so when you're talking about the nervous system, you're talking about its functions. Often we talk about one of the main functions of the nervous system is to send and receive signals. And so kind of, I guess, the workhorse cell that does that sends and receives signals are the neurons. So the neurons send and receive signals via electrochemical, I'm gonna write this out here, electrochemical, oh gosh, the writing is not great. Electrochemical signaling. So that essentially means that there's going to send an electrical signal that gets transferred into a chemical signal that turns back into an electrical signal. And we're going to talk about that pretty extensively in this chapter. But neurons are electrically excitable, and that's part of how they are able to work as signaling cells. They have cellular structures, so neurons are cells like any other cell. What's interesting, I think, is like when we were looking at epithelial cells, and now we're looking at neurons. They actually have exactly the same DNA located in their nucleus. It's just the way that that DNA is expressed that causes all of these different structural features that we're looking at here. So if we look at the neuron, you can see here, this is the cell body of the neuron. So this is where, when think about a cell, all of the organelles are kind of hanging out. You've got your nucleus, your mitochondria, your cytoplasm, obviously, your ER and your Golgi, all located kind of here in your cell body. When we look at the different kind of parts or structures of the neuron, you can see that there are these branches here kind of extending out from the cell body, and those are called dendrites. Dendrite is actually, I think, either Greek or Latin for the word tree, because they really do look like branches of a tree. Like even if you take a picture of a neuron from your brain, it's going to have these branching like projections called dendrites. And so dendrites are just basically kind of little signals, little, extensions of the soma reaching out into the environment around it, hoping to get some type of signal. So dendrites receive signals. So when it's reaching out into the environment, signals should be coming in from other neurons, sending those signals essentially to the cell body. Once all those signals come into the cell body essentially gets to decide, okay, go or no go. Are we going to send a signal or are we not going to send a signal? If it decides to send a signal, it sends the signal down this part of the neuron called the axon. Now this axon is one just long projection extending out of the cell body. And once that signal gets sent, that signal is called an action potential. Once that action potential gets sent down that axon, it's essentially a go processor. That signal is being sent. And once you reach the end of the terminal here, this is going to send out chemical messages from these axonal projections. Now, each one of these are going to be connected to the dendrites of another cell, and so on and so forth. So it's just kind of like this chain reaction of, this cell body with dendrites projecting out, sending the signal down its axon. The axon releases neurotransmitter, hits the dendrites kind of again give all the inputs of information to the cell body, and then that cell body has its own axon and decides whether it's going to send out that information. This is a very abbreviated kind of explanation of exactly what happens during synaptic transmission, because I know we're going to cover this later, but I can't help it. I get so excited. I just want to talk about synaptic transmission. So if this doesn't make complete sense to you quite yet, that's a-okay, because we're going to be revisiting it a few different times today. But this is, again, just kind of the general overview of what neurons do. Neurons receive messages, they send messages out. Their main function is to be the signaling cells of the nervous system. Okay, so neurons have a couple different structural compositions. And so we can see here that a multipolar neuron has these dendritic extensions that you see here, has one kind of axonal projection, and the information is kind of just flowing in that one direction. You can see that the axon terminal has multiple different outputs, but again, information in, message out. Okay, a bipolar neuron, interesting again. You have your dendrites kind of extending out. You can see that the dendrites are not connected to the cell body, so that would be one difference between the multipolar and bipolar neuron. This has, far-reaching kind of extensions or projections, and again, talking about anatomy and physiology, structure often belies function, and so this extension allows this bipolar neuron to maybe have dendrites extended way far out in a different region, have its cell body be located in one region, and then have its axonal projections in another region. And so that one single dendrite kind of out into that extension aids in the function of the bipolar neuron. And then lastly, you can see here a unipolar neuron where the cell body is kind of located outside of that whole system. And so you've got dendrites on one side, and then you have a peripheral process, and then we have the information being sent down the axonal terminal. So again, these are just essentially different kind of shapes or structures of neurons. I think the most prototypical type of neuron that we see pictures of are probably the multipolar neuron, but some of these other neurons are present in things like the peripheral nervous system. And again, their structure belies and aids their function in the peripheral nervous system. Something worth kind of mentioning here that we haven't talked about yet is the presence of this myelin sheath. You'll recognize that this myelin sheath is on all of these different neurons, and that they're kind of the myelin sheath is located on these projection areas, right? And what the myelin sheath does is it aids in conductivity, or it aids in signal sending. We'll talk about why that is when we get a little farther on, because myelin sheath is actually, generated by its own little cell. Okay, so like I said before, dendrite, again, in either Greek or Latin, I probably should look that up, means tree branching. And you can see here that there are different types of dendritic branching. So here is an example of two different types of dendritic branching, essentially styles. You have your Purkinje cell over here, and then you have a pyramidal cell here. Each one of these cell types is located in different regions of the brain. The Purkinje cell is located in your cerebellum, which is right down here at the base of your head. That's where your cerebellum is located. It almost is its own little mini structure tucked underneath your cerebrum. It has all of these dendritic projections. It's very important for movement. So this is important as you're moving, doing very delicate movements, that your neurons are able to talk to one another very quickly, and you're able to integrate a lot of information very quickly. And so here you can see kind of what those Purkinje cells look like. So if you took a slice of the cerebellum, you would see this kind of style of cell there. It's actually pretty cool. Pyramidal cell, a region of the brain that pyramidal cells are found in. There's a couple different regions, but one example would be like the hippocampus. The hippocampus is important for learning and memory. And so you have a lot of projections that kind of hit the dendrites up here, and then it sends information up the axon. So just examples of different brain regions that you find these cells. And again, just like we talked about in the previous slide, structure really belies function. In the example of the Purkinje cell, there's a lot of integrative information that needs to be brought into that single cell body. So you've got a lot of different small dendritic projections versus the pyramidal cell, which is still important for integration, but you're just not seeing the same density of dendrites in that case. Okay, so the functional classification of neurons can be classified in the direction of the nerve impulse propagation. And so, as I alluded to when we were talking about the peripheral and central nervous system and how we take an input from our environment and then we kind of integrate it into our brains and then we send out a type of response. This is where that nomenclature that I kind of alluded to takes place. So in the case of our sensory systems, things like our eyes, our nose, our mouth, our ears, again, these are all kind of sensory organs that have sensory receptors that hit sensory pathways. And all of those are our afferent pathways. So all of that afferent information, afferent going into the brain, into the central nervous system. And then to send out a response, let's say a motor response, you have to send out an efferent signal. So afferent in, efferent away. So all of those different sensory informations, again, as you're sitting there, maybe in your dorm room, you smell, let's say that person cooking something, they start to burn it and you smell the smoke and you're like, ooh, I don't know if that's a fire. So it's taking sensory information into your brain, you're processing it, and maybe your efferent response is to get up and go check on what's happening with the possible smoke smell, or maybe your response is to get up and exit the building because you smell smoke. So again, that's an example of your sensory system taking in the information up into the afferent pathway, your brain processing a type of response, and then activating some type of efferent response to that, which would be activation of your somatic nervous system or your motor movements. You do have interassociation neurons. They're also important for processing. So as we're talking about processing of information, even within your own central nervous system, there's a lot of association neurons there where maybe the smell of smoke reminds you of a campfire that you had when you were like 10, and that kind of triggers a memory. So it would be kind of like an association type of thing where you're not necessarily taking in the sensory information and creating a response. You're actually kind of cognitively processing different types of associations of that smell. So like, oh, smoke usually means fire. This reminds me of a fire I had, or even just that smoke equals fire is a type of association. Okay, so not belaboring the point too much, but again, this is kind of a nice simplified image showing this afferent and efferent kind of signaling. So again, you have your sensory receptors that have dendrites. So those are extending out here, and they essentially have that's a sensory receptor, not the cell body. So you have your sensory receptor that is essentially being activated by something. So if you have, let's say, a receptor in your mouth that is finds sweetness, and you eat something like something sweet, and it's going to activate that sensory neuron. Again, this is usually a unipolar neuron. It's going to go from that nerve body. You have enough sweetness in your mouth that's going to be sent to your brain in the central nervous system. At that point, you have some type of cell that gets activated, an interneuron that's usually multipolar. So you can kind of see that multipolar structure there. It's going to send another nerve impulse to another nerve. And at that point in time, here you have a nerve impulse that's sending out some type of response. So let's say you eat something sweet, it gets sent to your brain. Your brain's like, ooh, I'm super hungry. I need to start to produce saliva. So kind of, again, this is not necessarily a somatic response. This is more an autonomic response. So your body starts to create that saliva, and then that sends out a signal to some of your effectors. So things like glands just start to secrete that saliva, saying, oh, I'm going to start eating, so I need to secrete some saliva. Again, your glands don't necessarily cognitively process it like that, but once they get activated, they start to respond in the way that the upstream activation tells them to, which is to produce saliva, because we're going to start eating and I've just eaten something sweet. So the whole, digestive process needs to start. Again, that's an example of your autonomic system getting activated and not necessarily your somatic. Again, somatic is muscles, and autonomic would be something that's automatic. Things like glands that would be secreting saliva. Okay, so hopefully that example made sense. And if it doesn't, just please let me know. Okay, so types of neurons. Again, if you are a visual person and you like videos, it's a great example to check it out. Okay, so neurons are one type of cell that's found in the brain, and kind of the other type of cell that's found in the brain are called neuroglia. Glia actually, again, either Latin or Greek, means glue. So when glia were originally named, they were considered the glue of the brain. So here it says that they're not electrically excitable. I know people will probably beg to differ, but for in all intents and purposes of this textbook, they're not electrically excitable. They make up about half the volume of the nervous system. They can multiply and divide. They very often act as support cells to neurons, and they aid in neuron function. So they're kind of like the backup singers for the neurons that are taking, singing and being kind of the star of the show. Glia support them and make sure that they have everything they need to function properly. There are 6 different kinds of neuroglia. We'll talk about those 6 different kinds at one point. There are 4 different kinds in the CNS, 2 different kinds in the PNS. But again, they make up kind of the neural glue of the nervous system. Okay, so if we look at some examples of neuroglia, one example of neuroglia is the oligodendrocyte. Okay, oligodendrocytes, what these guys do is you can see here, you've got a cell body, and then you can see them projecting onto the axons of the neurons. And what they're going to do is they're going to essentially aid or not produce. They're going to supply the myelin for the neurons. Again, the myelin or the myelin sheath is very important for sending signals. It aids in conductivity, meaning if you send a signal, it conducts quickly and more easily through something that has high conductivity, which myelin does. Next type of neuroglia that we'll see here is the microglia. Microglia are like the immune cells of the brain. The nervous system is actually completely independent from our immune system as we kind of classically think of it. And so the brain actually has to kind of have their own immune system that that originates there. So the microglia are kind of, it's the brain's own version of an immune system. And then we have astrocytes. Astrocytes, again, kind of look like star-like projections. You've got fibrous astrocytes, protoplasmic astrocytes. Here's another protoplasmic astrocyte. And I'm not going to necessarily make you know the difference between a protoplasmic and a fibrous astrocyte. Just recognize these astrocytes have star-like projections. They are very much the glue that holds the brain together. These are very much workhorses of the brain. Part of what they do is kind of buffer ionic concentrations around the neurons. They also aid in uptake of excess neurotransmitter. So we'll talk about kind of the glial neuron interactions a little bit more when we talk about synaptic transmission, but they're definitely hanging around helping out the neuron carry out its function. Again, we have microglia, and then kind of the 4th type of neuroglia that we're talking about in the CNS is the ependymal cell, and this is going to be the cell that lines the ventricles. And again, this is just important for generation of cerebrospinal fluid. So the fluid that kind of bathes our brain and has all of the nutrients and glucose and ionic concentrations that the brain needs to function. The ependymal cell is essentially going to do that because they're lining the ventricles, which are these huge gaps in our brain that create this fluid. So this cerebrospinal fluid, again, the cerebrospinal fluid is just that fluid that bathes our brain. It just makes everything all right and gives the brain everything that it needs. Okay, so just to kind of, again, make it very clear, the different cell types. We've got 1 oligodendrocyte, 2 microglia, 3 astrocyte, and then 4 ependymal cell. And I kind of talked about the function of each one of those. So if you have any questions about that, though, feel free to ask. You can tell that I get a little taken away in describing things. Okay, so neuroglia of the peripheral nervous system. So we're talking about the peripheral nervous system and not the central nervous system. There's these 2 different types of neuroglia. So here you can see the Schwann cell. The Schwann cell is going to essentially create that myelin sheath. Again, that myelin sheath is so important for conducting the rate at which or the speed at which these messages are sent. And so you want to have a high rate of conductivity. And so it assists in kind of supporting that myelinated sheath. So here you see a myelinated axon. The Schwann cell here, you can see there are instances of unmyelinated axons as well. And then here you can see that satellite cell, again, just producing support for the neuron in a ganglion, which again, we'll talk a little bit more about what that means when we talk about the peripheral nervous system and kind of how it's situated. So neuroglia in the PNS, think about Schwann cells and satellite cells. Okay, so the myelin sheath is produced by Schwann cells and oligodendrocytes. So myelination. What's a great way to describe myelination? So myelination is in the conductivity. I keep saying that, but a couple different ways to think about this is right now, as I look, this is a wire that's sending, sound from my little speaker here and giving me audio, but you can see that it is covered in plastic that allows that signal to not be lost, right? Because there's electrical, signals being sent through the wire that's inside of this, but because I've got this nice plastic covering on it, one, I'm not losing the signal because it's not dissipating into kind of the world around it. And so essentially, myelin sheaths do that same thing, is that they kind of keep the signal kind of within that region. It's not dissipating to the environment around it. The other thing that it does is because it's kind of made of this fatty substance around the axon, it aids in the speed of conductivity. And probably the best way to describe that is, have you ever been swimming in a swimming pool and you get completely submerged and somebody like does something or you hear something scratching and you can hear it much more clearly underwater than you can kind of outside of the pool? And it's again just because of the speed of conductivity of water versus the air. And so again, there's an additional kind of speed of conductivity in the fat cells of the myelin sheath that's faster than even the extracellular solution surrounding it. So it really just aids in speeding up the rate at which a signal is sent down the axon. So unmyelinated neurons generally signal much more slowly. Myelinated neurons signal much more quickly. Again, that myelination aids in the rate at which a message is sent. And some some degenerative diseases are due to demyelination. So you lose that myelin, you're not able to send signals as fast, and things just don't operate the way they're supposed to. This is just kind of a nice image showing myelination versus an unmyelinated axon. Here you can see the Schwann cell surrounding it. Neurolemma, again, not a super important word that you'll need to know. But again, I think it's just showing you that nice myelination around the axon. Just an example, nice covering surrounding that axon. Okay, so we're talking about myelination. And maybe you've heard of things like white matter and gray matter when we're talking about brains. And myelination is directly related to white matter and gray matter. So if we're looking at the essentially transverse plane through that, here you can see the frontal section of the brain and the transverse section of the brain and the frontal section of the brain. You can see this white matter that's kind of projecting in and out here through all of these different regions. These are actually going to be regions of myelination that you see here. So all of that white matter is areas of myelination. And then if we look at the gray matter, those are unmyelinated neurons. And so those are generally going to be kind of the cell bodies. These regions that you see here, because they're kind of sending signals through and across the brain, it's very important that that happens very quickly. So it's almost like you have these freeways through the brain to send messages that are all myelinated. And so kind of those freeways are considered to be the white matter, the myelinated matter, and then the gray matter is going to be that unmyelinated matter. Those are more of the cell bodies than the axonal projections. And so again, they're kind of like freeways to the brain, and you want those freeways to function as quickly and smoothly as possible. So they're going to be myelinated. Okay. Electrical signals in neurons. An overview. So just pre-warn you, electrical signaling in neurons is very much the thing that I enjoy talking about the most because I'm an electrophysiologist by training. So I'm worried for you. Okay. So electrical signals. So cells communicate via these electrical signals that turn into chemical signals that turn into electrical signals again. And we're going to talk about how these electrical signals are sent. So when we talk about electricity, I think it's important to recognize that electricity is mediated by movement of ions and cations, and we'll talk about the movement of those ions. These cells essentially have um, when I talk about ions and cations, so we essentially are talking about the movement of things like sodium and potassium and chloride. These are probably the three most important cations and anions. The only other kind of divalent cation worth recognizing is calcium, but we'll get to talk about that more when we talk about synaptic transmission. But in terms of sending electrical signals, these three little guys right here are the most important to recognize, and we'll talk about kind of the movement of these. When we talk about a nerve cell, I often like to think of the nerve cell. Oof, what are you drawing? I think of the nerve cell as a salty banana, and we'll talk about this more when we're talking about the electrical changes that happen in neurons. But I call it a salty banana because there's a lot of sodium and a lot of chloride outside the cell. And bananas, if you know, have a ton of potassium in them. So this is just kind of something we'll revisit again and again, because it's going to be the concentration of these ions, either inside or out of the cell, that allows for electrical signaling to take place. So think of the neuron as a salty banana, meaning there's tons of sodium and chloride outside the cell, and there's lots of potassium inside the cell. So, kind of devolved. Okay. So in terms of the types of responses, so let's say you have, many of you probably have learned about action potentials, right? If we look at an XY axis, and this is kind of voltage again, because we're measuring electrical, we'll just write a V there, because we're measuring electrical changes. Your X is voltage, or I'm sorry, your Y axis is voltage, your X axis is time. Usually in the milliseconds. Many of you probably have learned about action potentials, right? The action potential. The action potential is essentially the go or no go signal for a neuron to be sent. Today we'll probably be talking about something called graded potentials. And so graded potentials are little depolarizations, little bumps in signaling that are kind of below the threshold of an action potential actually firing. Hopefully I didn't use too much vernacular there that we haven't described. But again, graded potentials are just kind of little bumps, little signal bumps, and actual potentials are just like, oh, we're going to scream and send this information on. Sometimes the way I describe it to students are, let's say you yourself are a neuron and you're having to decide whether or not to pass on a rumor that you're hearing. And so when you're getting this information from one person, you get a little bump of information, right? That's a little graded potential. Let's say you have two people who tell you the same thing, but you haven't said anything yet. Those are like kind of two graded potentials. But then let's say you have 5 people tell you the exact same rumor at the same time. And you're like, oh my gosh, I have to tell somebody. That's you essentially deciding you're going to send a signal, aka an action potential. At that point, your threshold has been reached and you're going to send that signal or tell somebody else that thing. So the graded potentials are essentially the cell getting input but not sending anything out. And then in the case of once it's reached threshold and it's heard enough signals, it's going to pass that on via an action potential. So we'll talk about this in several different ways. So production of an AP or graded potential upon existence of a resting membrane potential and the existence of certain ion channels. So we'll talk about this more in subsequent slides. Okay. So resting membrane potential, like I kind of alluded to before, the neuron is a salty banana. Okay. Trust me, there's a reason we're going over this multiple times. So the salty banana of the neuron essentially has far more positive charges outside the cell than inside the cell. So you've got far more positives outside the cell than inside the cell. So because of that, most neurons are resting at a negative kind of electrical value. So if you put, let's say, a huge electrode into that cell and recorded what exactly that was at, probably find those cells are sitting at roughly minus 50 millivolts. Now, this is important because you can say, okay, well, the inside of the cell is negative. And let's say if like a little doorway opens here, what's going to happen? What's going to happen is that you're going to have positively charged ions that want to come into the cell. So you're going to have these sodium ions that really want to come into the cell. And so they're going to come into the cell and essentially make the cell more positive. So not as negative, a little bit more positive, just a little. But again, we're going to talk about this numerous times. So we talk about resting membrane potential. We're talking about the cell when nothing is happening. And the cell resting membrane potential is generally negative, again, because you've got far more salts outside of the banana than potassium inside the banana. Okay. So when we're looking at examples of graded potentials here, you can see a sensory receptor that's being activated by the pressure of a pen, sending a graded potential up to that sensory neuron, all the way to the inner neuron that is located in kind of the base of your spinal column that gets sent all the way up into your brain. So what's interesting to note is that you're getting kind of a graded potential sent in, but then you have a action potential. So your afferent information is a graded potential, but your efferent response is an action potential. So you can see, again, because the action potential is black, you can see this cerebral cortex neuron has decided, okay, we've processed the information and now let's respond. So it's sending an action potential kind of down through the spinal column back out into the hand, again, activating the somatic nervous system. And here it's turning back into a response at the muscle. So let's just go ahead and go through this one more time. What's the best color here? I mean, maybe brown. So again, you have a sensory receptor here, graded potential being action potential being sent all the way up to your brain, and then once it gets up to the brain's processing it, and then you have a cerebral cortex neuron that says, okay, we got to respond by generating movement, and so then it comes back through your spinal column, and you can see it goes down and it innervates your muscles, and at that point it sends neurotransmitters to your muscle, causing your hand to move. So as you're taking notes, this is all happening in your hand, which is in your body, which is pretty amazing. And that is just one tiny little possible neuron or collection of neurons that's aiding you and allowing you to actually, draw something or take notes on your page. Again, this is your afferent pathway, and downstream is your efferent pathway into your somatic system or your muscles. Okay, so membrane potentials, excitable cells, and resting membrane potential. Okay, so we talked about resting membrane potential as a salty banana and you having far more positive ions outside the cell than inside the cell. So I briefly mentioned that there are little doorways that open for the ions to move in and out of the cell. And those little doorways are really called ion channels. And so ion channels, and when we think ion, we should be thinking sodium, potassium, chloride, and then, well, I'll just keep throwing calcium up there because we'll talk about it eventually. So these are, again, they're the four kind of ions or cations that you need to remember. Just these four. They're probably the most important for this whole process. Okay, so you can see that there are leak channels that alternate between open and close. So again, because the neuron is a salty banana, you've got more potassium inside the cell. So when that cell opens, things want to go from an area of high concentration to an area of low concentration. So always think about, I always like to think of like ions or whatever being kind of, and no harm to antisocial folks, but I always think of them being like the antisocial folks at a party who are just like, I want to get out of here right now. Let's leave. Like, I don't want to be here. There's too many people here. Let's leave. And that's pretty much the way potassium feels all the time. Potassium's like, there are too many other potassium ions in here. It's just too busy, like, let's get out of here. So when you have the random opening and closing of these leak channels, you have potassium leaving, and as that potassium leaves, it's continually making that inside of the cell more negative, okay? So potassium channels are more numerous than sodium channels. Again, this aids in helping the cell remain more negative than positive. So resting membrane potential, we keep talking about that. The resting membrane potential is always negative, always, always negative. Okay, if we look at ligand, so that's an example of a leak channel. An example of a ligand-gated channel. So ligand, think of a ligand as kind of like a key. A ligand's like a key that unlocks the door that allows the channel to open and for ions to move through. So a ligand can be a series of things. And in the context of what we're talking about right now, most often that ligand is going to be a neurotransmitter. I'm just going to write NT here because my handwriting is terrible, but you can see over here the example of a neurotransmitter. So a neurotransmitter is going to bind that ligand-gated channel. It's going to cause that ligand-gated channel to open. And at that point, that ligand-gated channel is going to allow ions to move. Now, that ligand-gated channel doesn't have to be, let's say, permeable, or doesn't have to let every ion through. Maybe it only lets some types of ions through. So in this case, let's say acetylcholine, which is a neurotransmitter, binds to the receptor or the ligand-gated channel, and that opens. Here you can see that you have potassium kind of going outside the cell, and then you have things like, let's say, sodium or chloride going inside the cell. So it just kind of depends on what that channel allows to go inside and out. So channels can be very selective, meaning it doesn't have to let every ion or cation in or out. And again, the direction at which those ions move is a direct relationship to where the higher concentration is. Generally, most instances, you're having them go from areas of high concentration to low. So that would be the case for potassium. But let's say if this was permeable to sodium, sodium is high on the outside. And so it would want to go in to where there are low concentrations. So that's kind of the way that sodium would want to flow. Okay. So think of sodium always wants to go in, potassium always wants to go out. The opposite of what the salty banana originally kind of showed you. Okay. Mechanically gated ion channels. Mechanically gated are opened via some kind of mechanostress or some type of, essentially, it's kind of bulldozery, but kind of literally some type of mechanical disruption to a system. So a great example of mechanically gated channel are the receptors that mediate touch. The way that they mediate touch is because they sense the pressure changes as you are touching something. The mechanoreceptors change their shape, and because they change their shape, it allows the ions, the essentially door to fly open. And at that point in time, you can have things kind of pass through. So the examples that we're seeing here is that we have a mechanically gated channel. You've got some kind of mechanical stimulus. As that mechanical stimulus happens, then you have the ions flowing into the cell. And again, if we remember, there are high concentrations of sodium outside the cell. And we haven't talked about the concentration of calcium, but calcium generally is much higher outside the cell. Calcium, if you're going to learn any more about cell biology, just recognize that calcium is just controls a ton. And because of that, and it works intracellularly inside the cell a lot. So generally, intracellular concentrations of calcium are kept very low, so it can be used as a signaling molecule. So intracellular calcium is low. Low intracellular calcium. So that's generally the way things are. So because of that, if a mechanoreceptor opens, calcium is going to flood in. Okay. So I would say just at some point, just drawing your little salty banana and start to draw out which ion is high. And we'll kind of do that, I think, at the end of this lecture. Okay. The last, I guess, type of ion channel in neurons that we'll talk about are voltage gated channels. And those are going to respond directly and changes to membrane potential. So again, our resting membrane potential might be at -70. Again, that's if you have a cell, you put a little electrode into it, you're measuring kind of the electrical inside what it is. And so there's essentially, well, that's probably not the best way to represent it. There is just like, let's say 2 little positive ions in here, but a ton of positive ions on the outside. So because of that, relative to the outside of the cell, because that's kind of your basis of comparison, the inside of the cell renders itself very negative, and so in this case, -70. So let's say you have a few of those positive cations that come into the cell that can raise that internal voltage to -50. So in that case, what's going to happen is you're going to have a voltage gated ion channel open, and that can allow a passage of ions through that channel. So probably the best example of that is the voltage gated potassium channel. So let's go ahead and draw a picture again here. So this is voltage, this is time. So we're going to just draw a quick action potential. So we got a small little bump here. This is essentially threshold. At this point in time, you've got a huge opening, and then right about roughly in this region here, the voltage becomes same. If it's sitting originally at -70, once it gets to -50, the voltage gated potassium channels open, and potassium's flowing outside of the cell. So it's going to make the cell more negative again. So that's what contributes to that decrease down there, is this is voltage gated potassium channels. And again, that voltage gated potassium channel opens, potassium rushes out of the cell, and the cell becomes far more negative again because you've got positive ions leaving the cell. I just really want to talk about action potentials all day, and hopefully this doesn't confuse the hell out of you. Okay, so the 4 different ion channels that we talked about. First were leak channels. So when leak channels open, that ion is going to move down its concentration gradient. So if you had a potassium leak channel, it's going to leave the cell. If you had a sodium leak channel, it would go into the cell. So again, these are found nearly all cells. They randomly open and close. Ligand-gated channels, those only open when the key or ligand is present. Often the ligand is a neurotransmitter. So we talked about acetylcholine as being the neurotransmitter. So once that neurotransmitter binds to that gate, it opens, and then again, those ions are going to move down their concentration gradient. So if you had a ligand-gated ion channel that bound your neurotransmitter and it opened and allowed passage of sodium, sodium would go into the cell. Again, salty banana, salt wants to go into the cell because that's where the lower concentration is. Mechanically gated channels, again, if you have some type of disruption to an area, so mechanoreceptors on your skin are probably the best example of that. They open, example, in response to mechanical stimulus. Touch, pressure, vibration, tissue stretching are all examples of that. And those are in the dendrites of some sensory neurons. And then lastly, voltage gated channels open in response to a voltage stimulus. And these are in the axons of all neurons. So when we talk about action potential generation, which we will in our next lecture, we'll talk all about voltage gated channels. So this is just kind of an introduction to what types of voltage gated channel or what types of channels there are. And then we're going to talk more about where exactly these channels function and how they help synaptic transmission take place. Okay. Okay, I'm going to come back to this slide very quickly. So again, you have a cell. I'm just going to draw this one more time for you, and we will talk about this more in the next lecture. Okay, so this is a neuron. And again, think of the neuron as a salty banana. So lots of sodium outside the cell, lots of sodium outside the cell, just a ton of sodium outside the cell, okay? And there is just a little bit of potassium inside the cell, but more potassium inside the cell than out. So we'll just go maybe like 4 of those guys in. Okay, the other ion that's important, chloride, lots of chloride outside the cell, lots of chloride outside the cell, maybe just like 1 chloride inside the cell. And then lastly, we'll go calcium. Lots of calcium outside the cell. lots of calcium outside the cell, and maybe just a little bit of calcium inside the cell, because there is some. Okay, now think about each one of these individually. So let's first start with sodium. Which way does sodium want to go? Sodium wants to go from an area of high concentration to an area of low concentration. So sodium wants to go in. Okay. Second, let's talk about potassium. Potassium wants to go from an area of high concentration to an area of low concentration. So potassium wants to go out. Okay. Next, let's talk about chloride. And chloride, we don't talk about it a ton, but just important to recognize still. Chloride, high concentration outside the cell, wants to go to an area of low concentration. So chloride wants to go in. And lastly, calcium. Calcium, area of high concentration outside the cell to an area of low concentration. So this wants to go in, okay? And the movement of these ions you might think is not that important, but next lecture we're going to talk about action potential generation, and knowing which way these ions move is just going to be super helpful. Like, just if you can learn nothing else, I think in this section, like knowing this, if you're going to take future neuroscience courses or understand neuroscience at all, super just helpful, really. Just knowing which way these ions want to go. Okay, so just take some time, draw this picture. And I think this is a great place to to leave off, because we're going to discuss this all, like I said, more extensively when we talk about action potential generation in lecture 2. And again, if you can't tell, super excited, very much love this subject. If you have any questions or concerns about it, reach out, because you can tell that you won't be able to shut me up, because I like talking about it. Thank you for all your time, and hopefully you have a great weekend. And yeah, that's all I have for you today.